everyone, my name is Celeste Dufour.
Hi, my name is Marissa.
Hi, my name is Christopher Warnicki.
Hi, my name is Matthew Culp.
Hi, I'm Alan Friday.
Hello, my name is Clara Scharnagel.
I'm Matt Chancellor, Collection Assistant in the Michigan State University Herbarium.
I'm an intern at the MSU Herbarium and undergraduate student at Michigan State University.
I'm a senior in the plant biology department. This is my second year working at the herbarium here.
I'm a graduate student here in the Department of Plant Biology at Michigan State University.
I am a postdoctoral researcher at the University of Tennessee in Knoxville.
I was the assistant curator of the MSU Herbarium until I retired 2 years ago, but I still work here as a research associate.
I am a postdoctoral researcher at the Sainsbury Laboratory in Norwich, UK. I did my PhD at Michigan State University and was based in the herbarium.
My name is Alan Prather. I'm the director of the herbarium, and I'm a faculty member in the Department of Plant Biology. This video is going to give you a digital tour of the herbarium and introduce you to what an herbarium is, what a specimen is actually consisted of, and how we use specimens in biodiversity science. Welcome to the Michigan State University Herbarium.
Hello all and welcome to the Herbarium Tour video. I'm Benjamin Mitchell, the digital media intern here at the MSU Herbarium, and I, along with the wonderful people you just met, will take you through a tour of our facilities, specimens, and we'll get to know some of the people that use and curate them along with examples of research we support.
The big picture is an herbarium is a collection of preserved plant specimens, so dead plants, not living plants, and those specimens are used in research. But the herbarium is really more than just a collection of specimens. It's really also all of the people that contribute to the herbarium. So at the MSU Herbarium, we have people that do things like specialize in stewardship of the collections and also do research in biodiversity science. We also have a great team of volunteers who help us with the stewardship of the collections, and students are involved in all aspects, everything from the stewardship of the collection to doing research. The herbarium was established in 1863 with a donation of about 20,000 plants to Michigan State University. 1863 was just about 6 years after the first classes at MSU, so that'll give you an idea that the herbarium and plant science in general has been really critical to Michigan State University since its inception, really. So I'm standing in front of a portrait of William James Beal. He was one of the early directors of the herbarium, and he did many things on campus, but among the many things that he did was to increase the holdings of the herbarium fivefold. So by the time he retired, the herbarium had over 100,000 specimens. Today, we have over 500,000 specimens, so we've quintupled yet again.
Great. Now that we know how the collection started and where it comes from, let's look at where it is today.
We've got about 524,000 specimens of plants, fungi, and algae, many of the oldest ones dating from the early 1800s, and we're also still collecting and adding new specimens today. By and large, most of our plants, the vast majority of our plants, look like this. So they're flattened, and they get a label down here in the corner that says what the specimen is. This is a sassafras. It's a plant that's native to Michigan, and we actually use it on our logo, so that's why I'm showing it to you here. We also have a very large and important collection of lichens that are mostly kept in packets, as well as a few other types of specimens like boxes or fluid-preserved specimens, and we even have some tree cores that were added just recently. One thing you might be wondering is, where did all these specimens come from in the first place? And it's really unique to each herbarium, because it depends on the research interests of the people who are part of the herbarium or part of that university or museum or institution. So many of our specimens come from Michigan in the Great Lakes region, Mexico, Borneo, Alaska, South Africa, and many infrequently visited places in the Southern Hemisphere, so Falkland Islands and other island chains in the Antarctic Sea that nobody ever goes to. If you care for these specimens properly, they will last hundreds of years. They're treasure troves of data freely available for researchers to use, now and in the future.
In addition to the numerous vascular plants, fungi, bryophytes, and algae that are securely kept in the MSU Herbarium, we also host a very special collection of lichens. And if you take the staircase up a level from the room Matt was just in, you'll arrive at just the place to see them.
So I'm actually now in the Lichen Herbarium, which occupies the top floor of the herbarium. It's massive. We've got 120,000 accessions, lichens, and a whole bunch more which still need proper sorting out. Herbarium is important for me, not just because of the size, but then where the specimens are from. They're from all over the world. We've got a massive collection from the Southern Hemisphere, and places like the Falkland Islands, the Greenland, Campbell Island, which very few people have ever visited. And because of this, the collection includes a lot of undescribed species. I have personally described, I think, about 50 new species from the collection. And I know there are at least twice that many still here which haven't been described. The collection is also important for me, for my research, as a source of comparative material. If I find a specimen, and I don't know what it is, I can just go into the collection, find something which I think is that species and compare it. So we think we've got a new species. What do we do? The first thing to do is to compare it to the other species in that genus, and always assume, of course, that you know which genus it belongs to. To do that, you have to look at the overall structure of the thallus, which is the body of the lichen, and also, more importantly, the fruiting bodies, which on this species are sort of round structures. And to look at those, you have to cut a section of them under the microscope, then say compare it to other species in that genus, which again is where this herbarium comes in. So I could go in, look at the collection here, and compare it to my species and be confident that it isn't the same thing. It's important to know what's growing, especially in a national park, because it is being managed for the conservation of the natural environment, and if you don't know what's there, you can't conserve it.
Supporting discoveries of new species is certainly a main function of Herbaria. However, our collections offer even more to students, researchers, and the world. Another traditional use of herbarium specimens is their role in floristics.
Hi everyone, Alan Prather again. I'm back now to talk to you about floristics. Floristics is the science of studying what plants grow where and why. That's one of the traditional uses of herbarium specimens, and really the history of herbarium specimens is that they were collected by people traveling the globe to bring back to essentially Europe, because this was hundreds of years ago, and they used those plants to understand the biota of the world. We still do those very same things with collections, and as an example, I will just show you the Michigan Flora. This is a book published that describes all the plant species, native or non-native, that grow in the state of Michigan. And specimens are basically the gold standards of documenting this kind of work. So you could rely on lists from 100 years ago, you could rely on observations, on things like iNaturalist, but the very best thing you can do is rely on herbarium specimens. And that's because herbarium specimens make science repeatable and reliable. Anybody can go back to an herbarium specimen to see if it was identified correctly. So basically, when the Flora of Michigan was written, every little dot on every little map is represented by a specimen in an herbarium. And the people that wrote that went to all of the herbaria in Michigan, including the one here at Michigan State University, looked at every plant we had recorded from Michigan, and used those data to build those maps, and also to discover which plants were growing in Michigan and which ones weren't. We still do those kinds of things pretty routinely. Another example is the Flora of North America. So this is a monumental work. It's going to be a 26-volume series describing all the plants that grow in the United States and Canada. And those treatments are written by scientists like me. So I've contributed 5 treatments in the Lamiaceae. One of those is the genus Monarda. So you can see some Monarda specimens here. The way that I use Monarda specimens in writing those treatments is multifold, but the primary things really are that I again had to document which species of Monarda grew in what states, and I did that using specimens. So one of these specimens of Monarda, for instance, was an unknown collection from Alabama that turned out to be the very first record of a species of Monarda from that state. When I discovered it and realized that it had been misidentified and checked and found that no one had ever recorded that species from the state of Alabama, I actually contacted biologists in the state of Alabama to let them know so they could make a record of that. That's how those things change. Another way that we use specimens in floras are that floras have to have keys. In other words, those are the couplets and the legs that you can use to identify a plant. And they also have to have descriptions of those species, so describing how long the leaves are, what color the flowers are, and those kinds of things. Specimens is where we get all of that information. They're the building blocks of floristics.
Specimens are also critical in identifying changes in environments. By preserving the past for future discovery, herbarium specimens are being used in new and important ways to support the evolving world of science and conservation.
I've worked with rare species, both here in the Great Lakes region as well as elsewhere in the country, and I'll be talking to you today about a few different ways in which specimen collections, and particularly the specimens that we have in the Michigan State University Herbarium, can be useful in plant conservation research. So specimens as a whole can be useful in plant conservation for a variety of reasons. One is that they provide a firm source of documentation for an individual population. So if I'm interested in finding populations of a rare species, say this Michigan monkey flower that's behind me, I can go to the herbarium, pull out specimens from the cabinet, consult the location that they were collected and the date and additional information that people have written down there, and then I can go into the field and try and find those populations again. This is an especially useful tool for rare species as populations are spotty and difficult to find by other means. Another way in which specimens can be useful in helping us understand rare species can be demonstrated by this Michigan monkey flower that's behind me. So to help us understand what is and is not a species, we often use different tools, some of which are genetically based and some of which are morphologically based, or like how the organism looks and is shaped and that kind of thing. So this particular species, the Michigan monkey flower, was once thought to be the same thing as a species of monkey flower that's found broadly to the west of Michigan, but doesn't really occur in this state. And some researchers, including researchers here at Michigan State University Herbarium, have looked into this species and done some genetic work using the specimens that we have in our collection, as well as that the morphological work, how the plant is shaped and the different proportions and that kind of thing, to demonstrate that the species is quite distinct from this other monkey flower. And so that helped us to understand that this is a unique thing that we have here in Michigan. The Michigan monkey flower is only found in the state of Michigan. That means it's endemic to this region, a word that sort of means only found within a given area. We have 3 plant species that are found only in the state of Michigan. One is this monkey flower, and the other 2 are in the sunflower family. Now, another way that herbaria and specimens are useful in documenting rare species is when a species vanishes from a region altogether. One of the ways that we know about it is through herbarium collections. So the species that's my background right now is the fringed puccoon. This is a species that occurs broadly just west of Michigan and then further west from there. And up until the late 1800s, did occur in Michigan. We know this because people collected herbarium specimens in St. Clair County in the late 1800s, and it since has never been seen again in the state. So we used to have this species in Michigan, and then it got lost as more development occurred and habitat changed and was lost, and the species vanished from the state. So that's called extirpation. It's a thing that happens with rare plants when they vanish from a given region. So it's one of the first steps towards its extinction. It's not quite extinct. It's lost from Michigan, but there are still populations of it elsewhere in the world. And we would never know about that were it not for specimens and the specimens that we have here at the Michigan State University Herbarium.
Herbarium specimens give us a record of when invasive and non-native plant species arrive for the first time. One example of this is garlic mustard. Garlic mustard is a newer introduction, first collected in Michigan in the 1900s. In fact, it was first collected in Michigan State University's Baker Woodlot in 2009, and this is the specimen that documents that. This is pretty important because forest fragments like Baker are refuges for hundreds of species, and garlic mustard is a serious invasive species that's able to invade undisturbed woodlands. Many other non-native plants can only gain a foothold in disturbed places. Our garlic mustard specimens serve as a benchmark to tell us how quickly invasive species spread and how they evolve in their new habitat.
It's not just individual species that change over time, though. Our habitats do as well, and specimens are key to learning about that change. Floristic surveys, where specimens are collected to document each species, can be repeated across decades to tell us whether species are vanishing, arriving, or persisting in their habitats. In Baker Woodlot, by surveying 40 years apart, we learned that species like yellow lady slipper orchids have vanished from the woodlot, and species like invasive garlic mustard have arrived. Those weren't the only species we collected, though. We found around 300 total. By collecting specimens, we discovered that nearly 20 native species have vanished from the woodlot, while 30 non-native species have arrived. The data seem to tell us that native species are being replaced in this nature preserve. It's a big deal because on an ecological timescale, 40 years is actually pretty short. This type of research tells us about how our plant communities are changing, which is especially important for bastions of biodiversity like Baker Woodlot, a high-quality remnant of Michigan's rich beech maple forests.
Noting changes of the past, researchers are using Herbaria for cutting-edge science that inspires brighter futures.
This year, I've been mainly focused on doing research on cobia with Dr. Ellen Prather. Cobia are a genus of flowering vining plants that are typically native to South and Central America. Over the years, Dr. Prather has constructed a phylogeny of this genus. However, in the past 20 years, since his last phylogeny, multiple new species have been discovered. Phylogenetic trees have been around for hundreds of years to show the evolutionary relationships between species. Typically, they've been constructed based on physical attributes, but since techniques to get genetic material have become more advanced and accessible, many people are reconstructing these phylogenetic trees to be more accurate based on their genetics. So here we have a specimen of Cobia pringlei from Mexico, which was sent over to us from an herbarium in Texas. This specimen came from the actual herbarium specimen, and from this specimen, I take about a full punch worth of organic material. I grind it up with nitrogen and run it through a process to extract DNA. From that, you get the product, which is just a clear tube of DNA, which we have to keep frozen. This we then run through PCR to get a tube that looks like this. So I have the product of the PCR here, and today I'll be running this through the gel to make sure it's viable. With the results of the DNA sequences, we'll be then running them through a computer to compare them to other DNA sequences to see where they wind up on the phylogenetic tree. So, specimens are important, especially their DNA, because they can help us determine whether a new species is present or where they evolved from and where they came from on the phylogenetic tree. You see, when we construct phylogenetic trees, we're really constructing where common ancestors are. Based on these common ancestors, we can see where these plants came from and where they evolved from. Matching that with different time periods or different cases of divergent and convergent evolution, we can see how these species evolve in the greater landscape.
So I study lichens. I'm a lichenologist, and lichens form a big percentage of the collection at the Michigan State University Herbarium, which is one of the reasons why I was very keen to do my PhD there and to work there, because that is a really valuable resource, particularly when doing biodiversity studies like I was doing. The specimen I would like to talk to you about is a herpethalin species, and this is one of the ones that I collected in the tropics. They actually have this fuzzy, almost felt-like appearance, and that is due to their structure as well as possibly due to some coating on the fungal hyphae of the lichen itself. And what this results in is actually creating a hydrophobic surface. If you put water on them, they'll bead into little water droplets. There may be potentially many reasons for this, but all of these are found in the tropics. They are found, particularly in my work, in lowland tropical rainforests, so places that get quite wet, quite saturated. And so it may be advantageous to not actually be fully inundated with water all the time, and thus to have this hydrophobic surface. And I wanted to highlight them for that reason, but also just because I find them really beautiful and really fun to encounter in the field. So before I get ahead of myself, I should say a little bit about what lichens are. Lichens are actually more than one organism. So classically, we have defined them as filamentous fungus and algae that live in symbiosis together. This, of course, is expanded to a filamentous fungus and a photosynthetic partner because it can be algae or cyanobacteria or both, filling that role. And now, in more recent times, we've discovered that there are actually additional partners involved, potentially, in the lichen symbiosis. And that actually brings me to some of the work that I was involved in at the Michigan State University Herbarium, investigating whether we could find evidence of this third partner in lichen specimens that already existed in the herbarium. So this was a project that was actually being conducted by an undergraduate researcher, Serena Whitner, who is now pursuing her PhD at the University of Hawaii. In terms of my own research and my involvement with the herbarium, I did my PhD at Michigan State University in the plant biology department under the supervision of Dr. Alan Prather, who is also the director of the herbarium. And I was interested in looking at patterns of lichen diversity. So we tend to, in many different groups, in flowering plants, in mammals, in reptiles and amphibians, we see the highest amount of diversity, the highest number of species at and around the equator in the tropics. And then as you go either north or south from the tropics towards the poles, you see a decrease in the number of species in those groups. But we hadn't looked at this for lichens. And specifically, I was looking at the lichenized fungi, so the main filamentous fungal partner in the lichens along this gradient, to see whether they in fact follow this pattern. But lichens too, it turns out, seem to follow a latitudinal diversity gradient. We have generally the highest species richness in the tropics and then decreasing species richness as we go north. And I had the wonderful opportunity to travel to many different places during my PhD to actually conduct a survey of lichen biodiversity myself. And all of those specimens are now housed at the MSU Herbarium. So they play a bunch of interesting roles, but they also are very hardy because they're able to live in all of these different places. And we've even put them up at the space station and expose them to, the radiation, the UV, the vacuum, the crazy temperatures of space, and they've come back essentially just fine. So there's really a lot that we can learn from the lichens in terms of survival and adaptation. There's a ton of potential in these collections, and I'm very happy to have been a part of them, to contribute to them, and to continue to work with them.
These specimens can surely bring us to infinity and beyond, but they aren't just for researchers. Here are some other ways herbarium specimens are used for classrooms and the community.
So we just got done talking about how specimens can be used for research. Of course, there are a lot of other examples besides just the ones that we went over in this video, but did you know that specimens are also great tools for teaching? They're sort of intrinsically relatable, as natural history objects. So the Michigan State University Herbarium supports MSU's teaching mission. We can bring specimens into classrooms. We can help students develop their projects. We can develop activities that are inquiry-based for students to do. And we can provide tours and in-person learning opportunities for students and others. We also participate in outreach. So keep an eye out for us at events like Darwin Day, obviously SciFest, and Fascination of Plants Day. We've worked with grade school classes and we've even visited libraries. So keep us in mind if you want your students to learn a little bit more about plants, because specimens are pretty great for teaching this stuff.
But Matt, how are specimens kept safe over so many years so we can do these cool things like research, teaching, and outreach now and in the future?
So someone has to take care of this huge collection, and it is a lot of work. That's what we call stewardship. We have a great team of faculty, staff, students, and volunteers who collect, mount, and restore specimens. This is so that future generations can use and build upon what this herbarium has become over the past 150 plus years. We manage stewardship by accepting new specimens and providing a home for them. And we provide a platform and resources for specimen-based research. So microscopes, field equipment like plant presses, stuff like that. One way that many herbaria like ours have been improving their stewardship over the past 10 to 20 years is through digitization. And all this is getting images of specimens and their label data into online databases that can then be searched. So when you can search hundreds of thousands of specimens at the same time, that really lets you start answering huge questions about biodiversity. A lot like Clara's lichen biodiversity project that she was talking about earlier. And so what you end up with is something really cool like this. And so if you ever wanted to access, we haven't digitized all of our specimens yet, but we've got about 300,000 of them into our various databases. You could go on our website under the digitization page and look there, and that's how you can access our specimens and download data and use them for whatever you want. They're there to be used.
This has been a tour of the Michigan State University Herbarium and a quick look at some of the exciting things that our specimens and facilities are used for. From aiding scientists in the field to supporting content in the classroom, the herbarium is here to help those with botanical ambitions. But where exactly is the herbarium?
The herbarium is housed in the Department of Plant Biology. That's in the Plant Biology Laboratories building on Wilson Road. And also, I hope that if you have any questions, you'll put them in the comments below or contact one of us so you can learn more. We're always happy to share. Thanks for your attention today.
This has been a production by the Michigan State University Herbarium. If you would like to see more, please follow us on Facebook at MSU Herbarium, Twitter at MSU_Herbarium, and like and subscribe here on YouTube. Your support helps us share our values of stewardship, biodiversity, and botanical awareness. And for that, we thank you.